Hi everybody! Today we're going to talk about RNA and protein synthesis. This is part of our big unit where we've been actually saying why does DNA matter, and we've been really examining the central dogma of genetics where, as you know, we've got DNA that we use as a template to make RNA, and then RNA goes ahead and goes through the ribosome and strings together some amino acids to make a protein, and those proteins are what make our traits, so it's kind of important. Today we're going to be focusing on this piece, the RNA part. Now, hopefully prior to this lecture, you have taken the time on Big Ideas, page 12, to review some protein synthesis, and we're going to go through those answers in a minute. By the end of the hour today, I'm hoping on your student tracker sheet that you can talk about learning goal number 5 and what you know about RNAi and its implications for current research and its applications. Also, I want you to get started thinking about learning goal 6, specifically transcription. The next piece, we're going to go ahead and turn to Big Ideas, page 12, and let's go through the answers that you reviewed. You know we are talking about protein synthesis, and we start with the DNA, and that DNA acts as a blueprint for making proteins. And the first step starts in the nucleus, and again, we're going to focus on that piece today. And the second step happens in the cytoplasm. We know that first we've got the DNA instructions, and DNA cannot leave the nucleus, but messenger RNA can, and so DNA gets transcribed over into RNA, and then that RNA goes through a ribosome, and that's where your protein is built. So, the first question when I was asking you to review on Big Ideas, page 12, is what are the monomers of proteins? And of course, those are amino acids. And then I asked, how do we put these basic building blocks together to make a protein? And that's through peptide bonds. What will determine the structure and function of proteins? That's the order and number of your amino acids. Then I hope that you wrote down some notes to yourself about proteins. Why do they matter in your body? And I hope you realize that your whole body is made up of proteins, your muscles, everything's made up of proteins. Even that little lens in your eye that's clear, that is made up of proteins. But we also use proteins to repair. All of our enzymes are proteins. When you grow, that's proteins. So it's super important. And the next question was, we know that mRNA only has 4 different kinds of bases. How can those 4 different bases code for all the different possible proteins? Well, first of all, those 4 different bases can be put in a different order. And then repeated, so you have lots of different combinations. They can also, once you make the amino acids from those different bases, those amino acids can be put in lots of different orders to produce all kinds of different proteins. So at this point in time, I'm hoping you've had a nice review of protein synthesis, maybe jogged your memory from some stuff from bio. Now, I want you to go ahead and turn to Big Ideas, page 13, and we're going to talk about the structure of RNA. First of all, RNA is ribonucleic acid. It is a single-stranded nucleic acid. It also, if you notice, has made up of nucleotides. They each have a sugar, they each have a phosphate, and they each have a nitrogenous base. Now, something to notice is their sugar in RNA is ribose, hence the name ribonucleic acid, where DNA has deoxyribose, or because the name is deoxyribonucleic acid. And another key piece to RNA is instead of thymine, like what we find in DNA, you're going to see uracil. And that's interesting. We're going to talk a little bit more about that in a minute. And overall, that was your structure of RNA. Now the function or the purpose is to take the instructions from the nucleus, from the DNA, out into the cytoplasm to the ribosome where your protein is going to be made. Now what I'd like you to do at this point is on your big ideas packet in italics, it asks you to compare and contrast DNA to RNA. I want you to do that. I also want you to take a moment to think about what base would uracil pair up with in RNA, and I also want you to think about why would we have uracil instead of thymine? Why does that matter? So work on that and pause me for a moment. Okay, hopefully you've taken a second to try to answer all the questions in italics on your own. And now let's kind of go through the answers. First of all, a similarity between DNA and RNA is they are both nucleic acids. They have that similar structure of a nucleotide that's made up of a sugar, a phosphate, and a base. Now, that's this piece right here. So here is our similarity. But if we go through these other 3 points, that's where we start to see some differences. So we know that DNA is double-stranded, but RNA is single-stranded. That's a big difference. Also, we've talked about the sugars. In RNA, the sugar is ribose, where in DNA, the sugar is deoxyribose. And this piece right here, uracil instead of thymine. Uracil in RNA, and thymine is what we get in DNA. So that means, what does uracil bind with or pair up with in RNA? But of course, adenine, the same thing that thymine binds with. But that gets us back to the whole question of why. Why does DNA, where all the other 3 bases are the same, but why does DNA have thymine where RNA has uracil? So I'm going to ask you to move to the next portion of their notes to ask that question. First of all, if you look closely at thymine, you'll realize it's very similar in structure to uracil. In fact, it's also known as a 5-methyl uracil, which means at position 5, it's methylated. Now, the interesting thing about uracil is it actually can pair up with any of our other bases. But when it's methylated, which means it's thymine, it can only pair with adenine. So now that you have this information, pause me and turn to your partner to talk about why this matters and why this would affect things. Hopefully you and your partner started to think about DNA and how important it is that we preserve the integrity of DNA. Because if you mess up your DNA, all the other things that follow that are going to get messed up. Specifically, let's say I pair something different with my DNA, every single protein that's made from it is going to get messed up. But if I mess up my RNA, only that one RNA transcript, the one time, will mess up that one protein. But from then on, you go back to the DNA, you make the RNA, and if the next set of RNA is fine, the proteins will be fine after that. So it's really important that we have thymine in DNA to protect the integrity, where uracil has that little bit of flexibility that wouldn't work well in DNA in terms of making sure generation after generation we passed on the same traits. At this point in time, turn your Big Ideas page and let's talk about the different types of RNA. So far we've been talking about mRNA. This is an uncoiled single-stranded nucleic acid that acts as our template. The DNA, again, cannot leave the nucleus, but the RNA can. So it gets the information from the DNA by the DNA acts as the template. The RNA then can leave the nucleus. It goes into the cytoplasm where it assembles those amino acids at the ribosome and makes those proteins. tRNA is also an RNA molecule, but as you can see right here, it has this T shape, which if you can picture this single-stranded molecule that is forms this T shape. And what it does is on the one end, it has an amino acid and the corresponding anticodon. So what tRNA does is it brings the right amino acid to match up with the mRNA molecule, because this anticodon on the tRNA matches up with a codon on the mRNA. The third type of RNA that we're going to talk about is ribosomal RNA, and this is in globular form. We don't know as much about RNA. We do know that it makes a way, it helps decode the mRNA into the amino acids, and it helps the tRNAs match up with the mRNA. So those of you who are my advanced learners who already know some of this maybe from AP Bio, you may choose at this point to spend some more time working with rRNA instead and researching where we're at right now with that. But today, we're going to focus on something that I find fascinating. It's called RNAi, or RNA interference. It's this really neat defense mechanism that our body has that is originally set up to prevent viral RNA from invading our cells and replicating. And so anytime an RNA sequence comes in that looks like it could be a virus, it is going to attack and stop that gene from replicating and making the proteins that go along with it. There are some interesting practical applications to this RNAi when we can sort of hijack this mechanism in our body and use it to our advantage. And you're going to watch a 15-minute video now from Nova that will help you learn more about this and hopefully see why it matters to us.